Hi everyone, my name is Nurul Zafira, and for today I will be carrying out an experiment on streaking for single isolated colonies. The streak plate method is used to isolate discrete colonies, and it requires that the number of organisms to be reduced. The number of organisms can be reduced by dilution, whereby the culture is streaked across the agar plate using an inoculating loop. When the culture is diluted, by streaking it across every quadrant that is drawn on the agar plate, the number of organisms will reduce. This allows the individual cells to grow sufficiently far apart from each other and ensures the separation of different species that is present. A pure colony can be obtained by picking isolated colonies and carrying out streak plate method on a driven agar plate. A pure culture is a type of culture that consists of only one type of microorganism that can be used for the study of cultural, morphological, and biochemical properties. A colony, on the other hand, is a group of microorganisms that is capable of growing on a solid agar medium.
Using a marker, draw a bisecting line on the bottom of the petri dish and divide it into 4 equal parts. Label each of part from quadrant 1 to quadrant 4. Sterilize the loop by flaming it until red. With sterile loop in hand, uncap the tube containing the culture and flame the neck of a tube. Take a loop full of culture and recap the tube. Turn the container over and place a loop full of culture by dragging it a few times across the quadrant 1. Use the edge of the loop and hold the loop at a shallow angle. Reflame the loop and allow it to cool, then turn the petri dish to 90 degrees and touch the area that has been streaked in quadrant 1 and drag it across quadrant 2. Reflame the loop and allow it to cool, then turn the petri dish to 90 degrees and touch the area that has been streaked in quadrant 2 and drag it across quadrant 3. Without reflaming the loop, turn the dish to 90 degrees and track the culture from corner of quadrant 3 across quadrant 4 using a wider streak. Reflame the inner coating loop to maintain aseptic technique.
The expected result for this experiment is that isolated colonies can be observed on the agar plate. From the image, we can observe that the number of organisms is reduced from quadrant 1 to quadrant 4. In quadrant 4, we can observe a single isolated colony formed on the agar plate.